event. This is our first event in a series of psychedelic science talks. Professor David Nutt is kindly giving us a talk on psychedelic science. So just a bit about Professor David Nutt. David was awarded an open scholarship to Downing College, Cambridge, and then completed his medical training at Guy's Hospital, London, and then continued training in neurology. He then completed his psychiatric training in Oxford, continuing as a lecturer and then as a Wellcome Senior Fellow in Psychiatry. In 1988, he set up the Psychopharmacology Unit at Bristol University before moving to Imperial College London in December 2008. David is currently Chair of Drug Science and President of the European Brain Council. And previously he has been a member and then chair of the Advisory Committee on the Misuse of Drugs. And he's also published over 400 original papers and a similar number of reviews and 8 government reports on drugs and 27 books. So I will pass you to Professor David Nutt.
So I hope I'm recording again, and it's a pleasure to be talking to you to your university celebration of psychedelic research. My name is David Nutt, and I am a psychiatrist and psychopharmacologist currently at Imperial College London. I've been working now in the field of psychedelics for over 15 years, and my talk today is going to look at the origins of psychedelic research, the setbacks and the resurrection that has occurred in the last few years. I'm going to start with this remarkable statement by William James, who's the father of American psychology, a hugely influential thinker and philosopher about the mind and consciousness. And in the 1890s, having experienced alterations in consciousness from drugs such as nitrous oxide and mescaline, he said this: our normal waking consciousness is but one special type of consciousness, whilst all about it, parted from it by the filmiest of screens, there lie potential forms of consciousness entirely different. And this is the most important statement. No account of the universe in its totality can be final that leaves these disregarded. And to my mind, that is one of the great challenges that have been thrown down to modern neuroscience. Consciousness understanding is a fundamental challenge to science. And of course, he then went on to say how to regard them is the question, for they are so discontinuous with ordinary consciousness. And in those days, they didn't have the tools that we have, particularly neuroimaging tools. And it's taken a century or more for us to really get to address the questions that William James raised. In that century, some remarkable things happened. Perhaps the most influential of all the experiences with psychedelics were those of Aldous Huxley. And you will all have heard of his book, The Doors of Perception, and I hope you've all read it. What you may not realize is that the title, The Doors of Perception, actually derives from a writing of a British philosopher and artist, a poet, William Blake. And Blake said in 1793, if the doors of perception were cleansed, everything would appear to man as it is infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. And what Blake was saying is that the human brain and the human mind is very constrained. And Blake was a visionary whose brain had broken free from the traditional constraints. And after Huxley took peyote, took mescaline, he realized that his mind could be changed by the drug. And he said this, based on his insights, he said, the fact that my mind has changed as a result of taking the drug means my mind has been constrained. And that must mean that the brain is an instrument for focusing the mind. And he's right. And modern neuroscience knows he's right. And what's remarkable about the 50 years on since he wrote this is that we have proved him right. So his insight turns out to be a very fundamental observation of how the brain works. LSD was made available in the 1950s. It was made available to researchers, particularly psychiatric researchers, but also it was used by interesting scientists. And these are two of them, arguably the most important Nobel Prizes in medicine and physiology ever. On the left-hand side, you have Francis Crick standing next to his model of DNA, and on the right, you have Kary Mullis. And Crick got the Nobel Prize for discovering the structure of DNA along with Watson. He helped Sanger get 2 Nobel Prizes for working out how DNA eventually leads to the production of proteins. And then after taking LSD, he began to realize that that was easy. Understanding the nature of life is easy. Understanding the nature of consciousness and brain function was very difficult. And he spent the rest of his life trying to do that. Kary Mullis, as a biochemist in California, was trying to get enough DNA in order to work out how it worked and how it differed between different animals. And he said that during an LSD trip, he saw the double helix molecule of DNA unraveling like a series of serpents in front of him, making multiple copies of itself. And he realized that that's what he had to do in order to be able to study DNA. So he hunted around, he found an enzyme, which we call polymerase. He used it to make multiple copies of DNA, and he got the Nobel Prize as a result of that. And he said that he would never have gotten the Nobel Prize if he hadn't had the insights that LSD gave to him. And I think both of those Nobel Prize winners would have agreed with what Einstein said. No problem can be solved from the same level of consciousness that created it. And that is why psychedelics actually are very important tools for understanding the nature of the mind and consciousness, because they change its function and nature. When LSD became available, as I said, in the 1950s, it was made by Sandoz in Switzerland, and they sent it out in physician samples in little glass vials to about 1,000 psychiatrists around the world. And it was used for 4 separate purposes. It was used to model psychosis. And we're doing that today. We're using psilocybin to make a model of psychosis on which we can test potential new kinds of antipsychotic drugs. It was used for self-experimentation by mental health professionals. And very often junior psychiatrists were given LSD on their early experience with their discipline in order to understand that their brains could change and to get insights into how their patients might feel when they suffer profound alterations of understanding and belief. And then it was used for therapy. It was used as psychedelic psychotherapy, largely in America, where they gave a high single dose, producing something like a mystical or peak experience. And this is what we do today with psilocybin in depression. But in Britain, another form of psychotherapy was conducted under LSD. It was called psycholytic psychotherapy. And their lower doses were given. This is a sort of micro or midi dose. So doses sufficient to break down some of the barriers that people have in terms of resisting attempts to engage in the therapy, or even subconsciously resisting accessing memories which are painful. And this could be used on a repeated basis so that people could eventually get more out of their psychotherapy sessions. And one of the pioneers in this field, and he's still around now, was Stanislav Grof. And he said this: psychedelics, when used responsibly and with proper caution, will be for psychiatry what the microscope is for biology and medicine, and the telescope for astronomy. And he's undoubtedly right, and he continued to work underground with psychedelics, and also other ways of altering consciousness that were less politically challenging, such as holotropic breathwork, when these drugs were made illegal. So if he was right and psychedelics are critical for psychiatry, why don't we use them today? Huh? And the answer is because the CIA in the 1960s were worried about American youth refusing to fight in Vietnam. And those young men who initially rather naively agreed to go to Vietnam to fight for America and the free world, as they were told it was necessary, realized that this war was a very unpleasant and eventually a pointless war. And here are two major quotes from soldiers who survived. Ron Kovic, you've seen the film, I hope, Born on the 4th of July, about him with his having his back, his spinal cord destroyed, being bound in a wheelchair. He said this: "All I could feel was the worthlessness of right here in this place and in this moment, all for nothing." And Michael O'Brien said, "I didn't want to die, not ever, but certainly not then, not there, not in the wrong war." And that kind of feedback from combatants in Vietnam led to the anti-war movement. And many young men who would have gone under the draft and to Vietnam, you could be 18 and you could be sent to fight just because your birthday ended on an even day or an odd day. They didn't want to go, so they went to San Francisco. They put flowers in their hair. They listened to Grateful Dead. They took acid and tuned in to the speeches and the writings of Timothy Leary there. But most importantly, they protested the war. And that right-hand photograph of someone saying, "Drop acid, not bombs," that was a reason LSD got banned, because it challenged the fundamental approach of America and to a large extent the rest of the world, that war was more important than peace. And people were terrified that taking acid would actually encourage people not to fight. And it certainly did. And eventually, psychedelics were banned. But to get a drug banned in those days, you had to have some evidence of harm. You don't have to do that now. In Britain, the Psychoactive Substances Act bans things that are psychoactive, even if they're good or positive. But in those days, you had to find evidence of harm. So how did you find evidence of harm from drugs like LSD, which weren't really very harmful? Well, you did what governments are good at doing, which is basically get newspapers to tell lies that suit the political direction at the time. And here are some clear fantasies of stories relating to LSD. And they're so self-evidently rubbish that no one actually remotely believes them. But it didn't matter, because as long as they were published, that was seen by the authorities as evidence of public concern. And public concern was then and is now a sufficient reason for banning a drug, even if the concern is fabricated by newspaper editors. And LSD was banned, and all the other psychedelics banned along with it, despite opposition from the most powerful man in the world at the time, Bobby Kennedy, Secretary of State. He would have been president if he wasn't assassinated. And he's saying this to his drug regulators. He's saying, why, if these clinical LSD projects were worthwhile 6 months ago, why aren't they worthwhile now? We keep going around and around. If I could get a fat answer about that, I'd be happy. Is there a misunderstanding about my question? I think perhaps we've lost sight of the fact that LSD can be very, very helpful in our society if used properly. He knew that the people calling for LSD to be banned, the CIA and the Drug Enforcement Agency and the FDA, were lying to him. But he couldn't stop the banning. And they were banned in 1967, and that ban was enforced in the 1971 UN conventions, and they've been banned ever since. And I would argue that this is one of the worst censorships of research in the history of the world. In fact, it's hard to think of a worse example. In fact, I genuinely can't, but when I look back over what censorship there has been for research over the last thousands of years, the nearest I can come up with is 1616, when the Papal Congregation of the Index banned all books advocating the Copernican system of planetary motion. Here you have in the middle Nicholas Copernicus, who, as you know, worked out that the sun, rather than the Earth, was the center of the universe at the time. And that conflicted with the teachings of the church, and they were very adamant that he was wrong. So they tried to stop his teachings, and anyone that promoted his teachings, like Bruno and Galileo, were either killed or basically forced to recant under the threat of death. That ban lasted 150 years. The ban on psychedelics now has lasted over 50 years. There weren't that many astronomers in those days, and the ones that there were, half of them were in Protestant Northern Europe, so they could carry on their research without impediment. So yes, it was a ban which extended around the whole of the Catholic countries, but it wasn't global. The ban on psychedelics for research has effectively been implemented in every country in the world, the 240 countries which have signed up to the UN Convention. And there was so much more neuroscience as could have happened in the last 50 years compared with the very limited number of people that were doing astronomical work back in those days. So I think the banning of psychedelics is the worst censorship ever. And regulators will say, oh no, we just banned recreational use. We didn't ban it for research. But in reality, they did. And this graph shows that very clearly. So the blue areas under the curve are the number of papers per year relating to LSD, and the red ones to psilocybin. And you can see that from the early 1950s, when it was made available, there was a massive increase in research. The US government funded over 1,300. In the decade from the 1955 to 1967, the US government funded over 130 separate grants on LSD. 1,000 papers were published, you can see if you add them up. 40,000 patients were in those studies. And the results were very positive and showing safe, effective treatments. And then when the ban came in 1967, people still had data to publish, so there was an output for a few years, and then it virtually disappeared. And it disappeared for two reasons. One is the US government never funded another study. And without that government funding, it's very difficult for any scientist to do research. And secondly, those who wanted to research it were effectively blocked because the rules that were put in place to access psychedelics are just so oppressive that it became virtually impossible for anyone to break through them. And very, very, very few studies, just a handful of studies were done from 1970 to 2015. I've written a couple of reviews about it. Here they are. The top one covers not just psychedelics, but also cannabis and ketamine, and the bottom one is largely about psychedelics. So please read those and see what you think. But we've been fighting back. My group and groups in the States, Johns Hopkins and New York University, and now other places have been fighting back. And I want to just share with you some of the work that resurrected science and also underpinned the new clinical research with psychedelics. And that was research we did at Imperial College with the Beckley Foundation. And we did the first systematic brain imaging studies with psychedelics. And this image here really sums up the key findings, which is that under psychedelics, the brain becomes much more connected. On the left-hand side, you see statistical connectivity in the normal brain under the placebo. And most of that connectivity is around the edge, and that's called the small world brain. Peripheral parts of the brain talk to each other. And obviously there's crosstalk because when you see something, you have to sometimes move in response to it. So you have to link visual cortex and motor cortex. But most of the processing of the brain is done locally in different regions of the brain. Whereas under psychedelics, this image on the right has the same number of connections as the one on the left, but you can see many, many more of them across the brain. And that opening up of connectivity in the brain is the reason, I believe, why psychedelics allow people to see things differently. They can tap into memories they've repressed, and they can come up with solutions to problems which they have failed to solve over the decades they've been wrestling with them. And what was so wonderful about that insight we gained from that kind of research is it completely supported the Huxley concept of the reducing valve. In doing so, the brain lowers what he meant was, in doing so, mescaline lowers the efficiency of the brain as an instrument of focusing the mind. He was right. One of the main purposes of the brain is to function in this small world way, to do fundamentally simple things as efficiently as possible, like working out how to get food in your mouth, working out where to go to get water, etc. That constrains the way we think because it makes the brain very efficient. But that constraint can be maladaptive. And if our brains are too constrained, then we can't solve problems. And psychedelics break down the ability of the brain to constrain the mind, and the mind can then do what it's good at, which is think and solve problems. And here is an example of how that works in terms of vision, and it's basically the way in which the brain sees things is that photons from an image, like that beautiful sunset, go into the retina. The retina then turns them into a whole series of time and frequency modified electrical impulses, which go into the brain, into the peripheral part of the brain, the back part of the brain, and the what and the where and the how are encoded in different parts of the visual cortex. And then the brain reconstructs an image of what it thinks you have seen. But that image is partly created in anticipation of what you've seen previously and what is out there, because that's the most efficient thing to do. But if you disengage those control centers, which is what psychedelics do, then you see things differently. Instead of seeing what you normally see, which might be a computer screen that is showing a slide like this, what you see are interesting, simple, but very powerful visual images. And we know from the physiology of the visual cortex that the classic images that are seen under psychedelics are generated in the very early part of the reconstruction of visual inputs. They reflect the very primary processing of the visual cortex before the simple analyses, the simple reconstructions are put together into complex full images. And that's quite remarkable. You can, under psychedelics, you can actually see these early workings of your brain, which you could never see really in any other way. How does it do this? Well, psychedelics work on the 5-HT2A receptor. These receptors are highly localized in the most evolutionary recent parts of the brain. You can see them here in the anterior and posterior cingulate cortices. They're in the parts of the brain which do the thinking rather than the sensory motor or the sensory integration. So they're strongly linked to emotional as well as other kinds of functions. And we know that these neurons are highly expressed on layer 5 pyramidal cells in the cortex. And the layer 5 pyramidal cells are the neurons which regulate cross-cortical integration. So the brain, in essence, is a two-dimensional computer. Each of these cortical columns is a computer, and the cross-talk between them is mediated through these layer 5 pyramidal cells. And they have high density of these 2A receptors, as do the interneurons, the chandelier GABA cells, which regulate them. And stimulating those 2A receptors massively disrupts the ability of these layer 5 neurons to integrate cortical function, and in particular to regulate what we call top-down function. The way in which the brain sees things is that you, as I said, you predict what's going to be there, and that prediction is done at a very high level, and it comes down to the visual cortex to essentially define what the visual cortex sees. But the visual cortex can't do that as well when the layer 5 pyramidal cells are not allowing the top-down so-called priors into consciousness. So then you see the primary workings of the visual cortex rather than the pre-constructed images that the brain expects to see. And based on this insight, we've come up with the idea that there may be two forms of consciousness supporting the initial musings of William James. And traditional consciousness, which most of us experience on a minute-by-minute basis, is the consciousness which defines the difference between being awake and asleep. It's driven by glutamate and opposed by GABA, and it relates to arousal, attention, memory, and parcellation. The remarkable ability of the brain to register and remember seemingly infinite amounts of data is all due to very specific activation of glutamatergic neurons, receptors in particular neurons. But under 5-HT2A simulation with psychedelics, things are very different. And in that, the psychedelic state, you've still got full activity of the glutamate GABA system, so you can still remember everything, but the content changes. You begin to bring in insights which are not normally part of normal consciousness. And those insights relate to the valence of the feelings, the meaning of the feelings, and an integration of sensations, such that people often have experiences like synesthesia. But that integration can occur at different levels. It can also occur at the level of emotions, not just in terms of sensory percepts, etc. So from that, we have now gone on to do research using these drugs to break down traditional priors, traditional precepts, which occur in disorders like depression. And James Rucker is going to talk to you about those. So all I need to do now is thank the team of people that have done all this work. Robin Carhart-Harris, who's led the team, Amanda Fielding and the Beckley Foundation, who gave us funding and supported us and drove us towards these remarkable discoveries. And I hope you've enjoyed the talk, and I'll be ready to answer questions at some point in the future. And enjoy the rest of your psychedelic session. Thank you.
Thank you, David, for coming to talk to us. Whilst this event was free, if you would like to support the charity Drug Science, then we will have more details over on our Facebook page. So that's everything from us. Thank you for coming, and you can head over to our social media to also hear about any future events. So thank you.